Okay.
Ready?
Yeah.
All right. Thank you. Good morning, everyone. Welcome to the Center for Clinical Trials and Evidence Synthesis Seminar. My name is Karen Robinson, and I'm a faculty member in the CCTES with primary appointment in medicine. And I'm particularly pleased to introduce the speaker for today, Dr. Daniele Wikoff. I met Daniele actually a few years ago. We were commiserating maybe about both serving on a National Academy of Sciences committee reviewing the EPA's IRIS assessment plan for inorganic arsenic. And that was a few years ago. Daniele is from Tox Strategies, which is a consulting firm where she's director of health sciences practice. And she also serves as a chair of the Science Advisory Council for the Evidence-Based Toxicology Collaboration, which is based here in the School of Public Health. Daniele conducts evidence syntheses, including scoping reviews and systematic reviews for human, animal, and mechanistic evidence streams to inform risk assessment. Her work uses and advances the use of evidence-based methods in support of hazard and risk assessment applications, including safety assessments for food ingredients and contaminants, industrial chemicals, and consumer products. And today she will provide an overview of evidence mapping and systematic reviews and risk assessments. Please welcome Dr. Wikoff.
Thank you. I'm certainly happy to be here and have the opportunity to tell you a little bit about something I really am quite passionate about. I will go ahead and share my screen. All right, can you see that, Karen? The slides coming through?
Yes, looks great. Thank you.
Thank you. Well, just a few more words to build on the introduction. Thank you very much, Karen, about the different hats that I wear in my day today. So my primary role is a consultant at Tox Strategies. We're a consulting firm of about 60 people, mostly toxicologists and exposure scientists. And we provide services both to the public sector as well as to the private sector. And through this, through my consulting work, I conduct a lot of evidence-based reviews of food ingredients or chemicals or things that might go into consumer products, such as cosmetics. I also wear a hat on the EBTC, both on the Science Advisory Council as well as the board, and have been involved for the last several years a lot with the education component and presenting at conferences, et cetera, to help kind of spread the word about evidence-based techniques. I'm also an associate editor for the journal Toxicological Sciences, which is the main journal. It's a journal of the Society of Toxicology. So also have a role in helping to disseminate systematic reviews within the toxicology world. So what I'm going to try to do today is to give a little background on evidence-based toxicology, then give some examples of how evidence-based toxicology is improving risk assessment specifically. I have a lot of busy slides. I will just tell you that up front, somewhat intentionally, but we'll try to give you the key points as I go. And would like to leave you with an appreciation for some of the continued challenges that we face in EBT as it relates to using systematic review and systematic maps and scoping reviews, et cetera, for toxicological questions. So first, just want to set the stage so we're all on the same page, thinking about what evidence-based toxicology is and how it might be, how it's different than evidence-based medicine. Starting with what toxicology is itself. Our fundamental principle is the dose makes the poison. And so this was really, Paracelsus is considered the father of toxicology, and everything we do is based around this concept that the dose makes the poison. There are a number of publications and efforts beginning, let's say in the, around 2004, 2005, talking about evidence-based toxicology and what is it really used for. And so I've just highlighted two papers here that kind of talk about what are we using evidence-based toxicology for. These focus on causation, and as part of that process, using evidence-based toxicology or a specific method or structured method to conduct what we typically think of as weight of evidence in toxicology. And so many entities, individual researchers started talking about this, maybe mid 2000s. So this is not as mature as evidence-based medicine. Some of the key differences when we think about EBM versus EBT, primarily that we're asking different questions. So instead of thinking about comparative effectiveness of an intervention or a drug or a nutrition measure, we're thinking about the potential for risk or hazard, or even a biological pathway. So what we've really observed is that we need flexibility in how we apply EBM type of approaches. So when we think about our structure of our questions, instead of using PICO, we tend to develop our questions in the PECO format, where we're looking at a certain type of exposure versus an intervention. And perhaps even more importantly, when we think about differences in evidence-based medicine, EBT, and again, this is more in thinking about how to use the existing methods, is that we're often looking at a different type of evidence base. So we almost never have controlled trials, as you can imagine. Chemical exposures, you don't have you don't have controlled trials of chemicals that might be adverse. Instead, we typically look at laboratory or experimental animal studies as well as in vitro studies. Sometimes we have observational studies in humans. That's probably more the exception than the rule. So when we think about my day-to-day, I look at almost exclusively animal data and in vitro data in conducting my reviews. So some of the pioneers of transitioning and developing methods for conducting systematic review for evidence-based toxicology was really the National Toxicology Office of Health Assessment and Translation, or OHAT program. So initially in 2015, they issued a handbook for conducting literature assessments. They also developed a risk of bias tool for human and animal studies. So this OHAT tool is one of the methods that many of us really use as a foundation for our assessments. And this is a little bit of a busy graphic, but I wanted to show it so you can see some of the similarities, but perhaps also some of the differences in how data might be evaluated. So the first steps: problem formulation, finding evidence, extracting evidence, assessing the quality or validity. Those are quite consistent with methods that you might already recognize. But when we get to some of the synthesis and integrating being the key step, and I have a subsequent slide to talk about that, we see some deviations from existing methodologies, for example, from Cochrane, on how to help interpret that data. In the inverse, again, looking at adverse effects. The OHAT program, in addition to having the risk of bias tool and the handbook, has also had a number of early applications that many of us look to for examples. And again, is focusing on hazard. So I have a blow up here of the bottom part of that diagram, and I think this helps to really put in perspective what some of our challenges are, is that we have to integrate evidence from different streams. So Karen introduced this idea of streams. We often talk about human data as a stream, animal data as a stream, and mechanistic data as a stream. And so we need ways to combine that to develop conclusions. And so here in OHAT, what you have is a matrix where you compare the level of evidence for health effects in human studies, that's on the y-axis, compared to the level of evidence for animal studies. And then you come up with a category for a hazard classification. Is this a known, presumed, suspected, et cetera, hazard in humans? So I'm emphasizing here that this is also, again, on hazard. So this is more of a yes, no, might this be a hazard? But my practice is really thinking about risk assessment. So I want to also give some background on how evidence-based toxicology can facilitate the risk assessment process. So before I dig in, it's also important to understand what exactly is risk assessment. And what I do is the practice of evaluating hazards and the dose response of that hazard. So how much of a chemical is needed to elicit a given response. In risk assessment, that threshold or that health benchmark that can come out of that dose response assessment is compared to an exposure. And that comparison of the health benchmark to exposure, which one is bigger, the potential for effects or your exposure, is known as risk characterization. So that is really when I typically do systematic assessments, my goal is to think about all of these components. So this is risk assessment is something that many authoritative bodies practice worldwide, thinking about food ingredient safety and feed ingredient safety, such as the European Food Safety Authority. The US EPA has been also a founding body when it comes to systematic review methods. And of course, they're thinking about chemical safety. That might be from the air, from different routes, from Superfund sites, many different applications. The World Health Organization is also utilizing systematic review, as well as a number of other agencies and scientists. But something that's really important to emphasize is we don't yet have a standard approach. We're really finding that we need fit for purpose types of approaches, which may not be unique, but we're still in the phase of developing those fit for purpose approaches for each of our given needs. So, for example, for a mandate to do something a certain way, to do risk assessment a certain way, let's say in Europe, might be different than in the US. And so those methods are still being developed as we speak. So kind of coming back to now, what is risk assessment and what methods do we already have? Think about frameworks. So Cochrane is a good example. Cochrane's very well established. Many people use it. We don't have that quite yet. So where are we? We have several frameworks that exist to characterize hazard. So I've already introduced OHAT as an example. Another is called NavGuide. There are also other entities that have developed approaches to think about hazard. We really only have two that have looked at the whole, thinking about systematic review to facilitate the entire risk assessment process. That's the US EPA TSCA program, which is a rulemaking body thinking about chemicals. And then European Food Safety Authority has also conducted a number of assessments that doesn't have guidance necessarily yet on the whole risk assessment process. There are also a number of approaches that have characterized how to apply systematic review methods from the hazard idea and dose response combination, which is really coming up with a toxicity value. And those are really what I'll focus on for the next series of slides. So in a publication from last year, this is a busy slide and I'll walk you through it. One of the things, we issued a publication that really is meant to address this lack of frameworks for conducting systematic review to facilitate risk assessment. And in this, we're not suggesting a specific, any specific way per se, but we're highlighting and helping to bridge how do you use systematic review to do your risk assessments. So we're targeting an audience where that usually conducts risk assessment, and the steps of that are on the right side of this slide shown in green. They really are quite similar to what's done in systematic review. But what systematic review really brings is additional structure to how you do each of those steps. The arrows in the middle are how to bridge that, and highlight some of the areas that are tricky when you are trying to apply risk assessment techniques using systematic review. So we've already identified, for example, the need to use a PECO-based approach. But what we look at in risk assessment is often quite broad, and I've got a number of examples to demonstrate that. So we need a way to go from broad to narrow for risk assessment. We also, I will highlight the systematic appraisal of quality. So quality has always been something we've thought about in risk assessment, but we've always thought about it very differently than risk of bias and internal validity. So that's a very new concept for most of our field. We also need to adapt some of the methods when we think about a grade-based synthesis of evidence or competence of evidence. That is helpful, but oftentimes we need to take things, look at things a little slightly differently, or perhaps in addition to a grade-based analysis, really focusing on things like dose response, mode of action, and uncertainty analysis. So this is another busy slide. It's also a figure from this manuscript, but it helps to now take another step. What do you really mean by this broad to narrow type of concept? So on the very left side with the triangle, this is a demonstration of really the risk assessment process. You're thinking about a single chemical or an agent, and typically we look at all of the data across any potential adverse outcome. Now, within those outcomes, let's say cancer, we might look at specific endpoints, so a specific type of cancer, liver cancer, brain cancer. Within that, we then hone in on specific data sets. And typically in risk assessment, you pick one or just a handful of studies to then develop that toxicity value. So that's a little bit different than, let's say, conducting a standard meta-analysis across studies to say yes or no, might this cause an effect. We need to actually select studies or a study in many cases to develop that toxicity value. So we have to go from lots of different outcomes, potential adversities, and start to narrow that down to these specific studies. And we do this slightly differently. The process and risk assessment is slightly different for cancer and non-cancer types of studies. And when we do this, we have to combine human, animal, and mechanistic data to make those decisions. And the mechanistic data in particular can be used in different ways. So the way that when I think about how systematic techniques or EBT techniques can be used is to first use systematic maps, where you broadly characterize the landscape of the topic or of the potential adverse effects, and then you use systematic review to do a more narrow evaluation and quantitative assessment of the data themselves. So getting a little bit more granular, what does this look like? Many of the steps here might look very familiar. You start with problem formulation. On the left side, you do your systematic map, and you think about the steps there. You develop a protocol, identify the evidence, do the map, reporting. And then from that broad scope, you can decide what outcomes or what potential adversities might I want to dig deeper on. And then you conduct your systematic review, shown on the right side of this slide. So the green steps, again, are quite general and overlapping. But where we see things differentiating are more when we get to the gray and the peach on the right side. So when we think about conducting critical appraisal or risk of bias, that is still very important. But one thing that we have found with toxicological data is we need to think about other types of validity. Here we've talking about construct and external validity, but of course there are many different types. And all of those are important to developing conclusions for hazard, mode of action, and risk. So now I'm going to move into some of the examples, and I've got 3 sections. The first here is talking about scoping reviews. I'll point out that the field uses these different terms to represent generally the same thing: scoping reviews, systematic maps, and literature inventory. So those are things to just kind of be aware of, that those terms are used somewhat interchangeably, but generally are doing the same thing. And I believe you all have a fairly strong background in evidence-based techniques, but really what we're talking about is characterizing a state of knowledge in systematic mapping or scoping reviews. And so you have more broad or open frame questions, and you don't go quite as far in the systematic review process often in terms of critical appraisal and synthesis. Compared to systematic review, you're using a much more closed frame or narrow question to answer direct questions or specific questions. So the first example is a publication with actually many systematic map examples that I think you might be interested in. So this was by Wolf et al. And published in 2019. And there are some snippets here on the right from some of the figures where essentially you're using a systematic process to conduct a literature search, but then you're using a number of visualizations to help characterize the landscape of information. And so here what they're looking at is a number of flame retardant chemicals that are very different in structure and function. And they're looking at the types of toxicity that have been evaluated in the literature. So reproductive, neurotoxicity, carcinogenicity, et cetera. And this helps to then characterize how much evidence and perhaps the type of evidence that is available that helps risk assessors to then decide what endpoints or outcomes might I want to further explore to help to develop my risk assessment. The next example is actually one of my favorites since we do a lot of research and support for sweeteners. So things like aspartame and sucralose and saccharin. So this is actually an example from Lohner et al. And it's a Cochrane-based group publishing in the Nutrition Journal. And this was a first step in conducting systematic reviews. And really what they've done is looked at what type of data are available for each of the sweeteners. And I like this graphic on the right because it combines the type of information with the potential adverse effects. So you've got blood pressure and CVD and diabetes, et cetera, with the sweetener shown at the bottom. And this is really helpful to then think about, if you need to evaluate the literature to develop a safe level, to know what kind of evidence that you have. Now, these authors also talked about the outcomes, the direction of the outcomes, for example, to then help also hone in on the potential significance of the adversities. So now moving into some systematic review examples, thinking about the apical outcome. And when I talk about the apical outcome, I'm talking about something adverse, something measurable at the end, something that you might observe, your doctor might observe. So something like cancer or a reproductive effect. I wanted to actually start with an example from the EBTC program from Hopkins. So this is one of their first actually forays into the systematic review process, and they certainly learned a lot along the way. And in this effort, the objective was to determine, it was essentially to compare a zebrafish embryotoxicity test with a mammalian prenatal developmental toxicity test. So you're essentially looking at a method, a new approach method, predictivity of a test that we have run for decades, essentially, when thinking about chemical safety. So could this be a potential replacement of those rat and rabbit studies, essentially? So they did systematic reviews of the zebrafish embryotoxicity test, as well as a systematic review of the rat and rabbit studies. One of the, lessons learned is the evidence base was really large. So they needed to hone down that evidence for 24 chemicals. So they were trying to say which chemicals have been evaluated in both types of tests. And then they compared the results and they developed conclusions, not only about the science, but here they were also able to develop conclusions about some of the strengths as well as limitations of the zebrafish embryotoxicity test conduct, the method itself, as well as some of the reporting areas that would really improve the field should these things be addressed. So the next example I have is a systematic review that we did on caffeine. And this was actually one of the first that I was really involved in, and it was a large one, as you can imagine. And what we were doing here is we were essentially thinking about the safe level of caffeine in a day. And we did this for a number of different populations. And we did this for a number of different outcomes. So really, you can kind of think about this as at least 5 systematic reviews. But the risk assessment objective is to think about all of these at once. So no matter how you slice and dice it, you can see that you're asking a lot of questions, but we've structured this for each population. We looked at caffeine intake above or below a given dose, and that dose was essentially the currently safe level. And we were asking the question, essentially, do the data still support these safe levels that have been proposed by some regulators? So we looked at all of these outcomes, and of course found that the synthesis and integration strategy is challenging for a large body of evidence. I know that's not news to any of you, but the goal was to really look at all of the available evidence. So on this slide, I've highlighted some of the approaches that we took. So in addition to characterizing the evidence that we had, we did that for each endpoint. So we had 27 endpoints within those 5 outcomes. So for example, within the bone outcome, we might have had bone density or different measures of bone measures. The same with acute toxicity, whether it be seizures or headaches. So we looked at different endpoints within each of those outcomes. And we tried to follow the systematic process from OHAT in doing this. In developing conclusions, one thing that we found important was to not only look at our confidence in the body of evidence, but also the level of adversity. We then developed conclusions by endpoint, outcome, and overall. So we gathered each of those, the data sets on the left. We made a little bit of a heat map, a little hard to see, but essentially the green highlights on the left side indicate what the body of evidence supported. Do they support the existing? Should it be lower? Should it be higher? What do the data support? So we looked at that in totality across the endpoints to develop conclusions.
Danielle, we have about 25 minutes left in our talk.
Perfect. Okay.
Thanks.
All right. I've had a lot of coffee today, which keys off the last example. So hopefully I'm not talking too fast. The next example I've got for you is a systematic review where we were looking at the dose-response relationship between dioxins during a sensitive window of development and reduced sperm count in rats and humans. So here we had a lot coming into this assessment already knowing that dioxins are hazardous. We know also that reproductive effects are some of the most sensitive. And specifically, we knew that effects on sperm were of critical importance. So we already had a lot of knowledge coming into this assessment. So our goal was to not ask the question, does dioxin cause reproductive effects, but instead, what is the dose-response relationship? And what's unique about this particular assessment is that we actually had a lot of data that looked at sperm count. So often in my day-to-day, we don't have 16 studies on a given endpoint. We have to combine across different types of measures and endpoints to develop conclusions. But here we actually had very similar measures of an outcome, and we were able to combine the data sets. They were still quite heterogeneous. However, the advantage here is we were able to quantitatively integrate that information to look at the combined dose response using meta-regression. So again, meta-regression, meta-analyses, not new in evidence-based medicine, but in evidence-based toxicology, it is a deviation to think about developing those health-based benchmarks from a combined big data analysis versus a single study. So this was unique. Instead of picking one study, we tried to look at everything together, the dose response as everything together. The US EPA has also been taking this approach. And this is actually where I met Karen. So I've been remiss not to mention this assessment. So I just have some screenshots on this slide that highlight what the EPA was doing here. And this was actually presented, I think, in 2019, where they were looking at using these Bayesian meta-regression techniques to do low dose evaluation, low dose extrapolation, to think about developing those toxicity values. And so they were thinking about this for a number of different endpoints over on the right, everything from different cancer types to pregnancy outcomes, skin lesions. So we're thinking about a lot of different endpoints or outcomes and trying to apply these techniques. I will also point out that this is a unique example in toxicology where we actually have a lot of epidemiological data. Now, this is all observational data from observational studies. But these were used to develop potential toxicity values. Okay, so now moving into systematic reviews for biological pathways and mechanistic data. And I'm going to spend a little bit more time explaining the projects or the examples here, because I think these really highlight some of the challenges of where we're at when we're thinking about risk assessment. So applying the evidence-based medicine techniques and approaches to more apical outcomes has been more straightforward. But applying these techniques to pathways, so biological or toxicological pathways and to mechanistic data, has been an incredible challenge. And I will just preface this with, we still have a long ways to go. So I'm going to give you some examples and talk about some challenges, but really want to emphasize, these don't encompass all of the work that's being done in this area by any means. So first, what are mechanistic data? And I think it's important to say it really kind of depends on who you ask. But when we think about a pathway, which is shown at the top, a biological pathway, we think about first of all, the molecular event. These are typically discussed as an initiating event. And that molecular event begins a pathway or a cascade of different responses at the cellular level, organ level, organism, and population responses. So as an example of different types of endpoints or measurements that might be done that we would consider mechanistic, typically, we're talking about the left side of this pathway. So something that happens at the molecular level or the cellular level, or potentially even the organ level. And this is important in risk assessment because typically we develop toxicity values based on that right side. So we would develop the toxicity value based on, let's say, an organism response, that cancer or impaired reduction. But typically the data that we have in an evidence base are mostly about this left side. And we need to use this information from the left side to help us develop conclusions about those apical outcomes. So we have different measures of DNA binding or cytotoxicity or a protein production, all sorts of different things. These can go up and down. They're done with lots of different assays. So we have a challenge in trying to combine these. So there's been a lot of work in particular to think about how to use systematic review methods to integrate these mechanistic data and pathway-based analysis. And we talk about these as adverse outcome pathways or AOPs or modes of action, MOA. Adverse outcome pathways are stressor agnostic. So the idea is you just have a pathway to a response that's characterized. And then modes of action are what we think about in risk assessment, and they're very chemical specific. And they help us make decisions about how we extrapolate data. So I've just got a conceptual drawing here, thinking about how a chemical can move through that pathway. But let's say we don't know all of the pieces. Maybe we know several of the pieces, but only one or two of the key events we think are missing, or possibly maybe just events at that level, that biological level organization might be missing. So we're finding that scoping reviews can be very helpful to start to identify key events or key event relationships to develop these pathways, at least putatively or initially. And then systematic review is more helpful when we already have a somewhat known pathway and we're trying to really specify or understand it even better. And so this is where we would use systematic review to really help us to understand the essentiality of each of those events, the domains of applicability, the dose levels, things like that. And those, again, are what's critical for decision-making in risk assessment. I will point out this is another opportunity as well. There are already guidance, like we have a lot of guidance as practitioners that we follow for developing these pathway-based assessments. So this is another opportunity where we're using systematic review to help us facilitate an existing process. So one example of this. So in this example, our PECO was that we had a single stressor and we already had a putative pathway looking at a specific outcome. And this was based on a publication by some EPA authors where they were looking at key events associated with trichloroethylene and cardiac adverse events. So the figure on the right is intentionally small. The idea is to show that through a review, systematic review, they identified potential events, but they didn't necessarily know everything. You can see some question marks on the left side. So we conducted a systematic review using this AOP-based approach to help synthesize and integrate mechanistic information. So again, this graphic is also intentionally small. You're not meant to read what's in the cells, but instead to understand how the data were organized, how we use systematic review to help structure our evaluation. And so what we have on the top level or the x-axis, we have those biological levels of events. So going from molecular to tissue and organ responses. And then on the left, we have data by species. So, or stream, if you will. In this particular evidence base, we had a lot of different species: human, mouse, rat, rabbit, chickens, zebrafish. So we had quite a diversity of types of models or constructs. And so what we did is we organized the data by these two parameters. And then we also indicated with the color coding and the shading, the direction of effects or support for that given event. Combined with the shading indicates how much confidence we had in that evidence based on risk of bias assessments. So the darker the shade, the more confidence that we had in that given response. And one thing that you can understand very quickly is that when we looked at mammalian models, so any data we had in humans, mice and rats and rabbits, is that they're dark, and those data were saying that they did not support that given pathway. But what we did see is when we looked particularly at models in chickens and zebrafish, there was more support for this pathway. So using this type of approach really added a lot of transparency and objectivity and also reproducibility. And it helped us to integrate study reliability into this evaluation of AOP. And it really helped us to look again at this pathway-based approach. However, when we did this, we really couldn't entirely follow off-the-shelf types of tools for systematic review, particularly when it came for appraisal, synthesis, and integration. Another area where we're seeing a lot of application of systematic review is in the evaluation of endocrine disruptors. And this is unique in a certain way because endocrine disruption is a mode of action or a pathway and not an outcome. So classification of a chemical as an endocrine disruptor is really significant. So this is really important in our field right now to be thinking about. The European authorities have a specific approach for identifying endocrine disruptors. There's a lot of boxes on the right side, as you can see. There's a lot of steps to evaluating and making these determinations. And the guidance indicates that systematic review should be used to essentially conduct this analysis. So seeing a little bit more about the top left of that graphic, that gathering of the information and using systematic review. The types of information that we're looking at are non-guideline studies as well as guideline studies. I won't spend a lot of time about that, but guideline-based studies are a pretty critical element in our practice. We're also going to pull in information from in vitro models and high throughput types of resources. as well as epidemiological data. Now, I've got a little bit of an X here because in this endocrine disruption guidance, the focus is really on in vivo and in vitro studies. And it doesn't give clear ways necessarily to say how to integrate that epidemiological data. But what this guidance does give us is a very clear structure for how to think about the potential for adverse outcomes. So you can see, again, we're thinking about biological pathways, and we're talking about the type of data that can inform those. So in vitro mechanistic on the left side, let's say, can inform those early events. And then we use in vivo mechanistic studies to characterize those intermediate events. And then we have specific study types that are designed to be sensitive or directly diagnostic of endocrine disruption pathways. And so we combine the data or integrate the data in specific ways. That epidemiological data or in silico modeling is considered a level 1, is essentially given the least weight. Then experimental data come in at the further pathways. And we conduct this process for each modality: estrogen, androgen, thyroid, and steroidogenesis, using lines of evidence and data sufficiency as well as data quality. The other really important thing that we do here is we integrate the data to determine if there's activity as well as adversity. So we have a very structured way to be thinking about this. To give you an appreciation for how complicated this can get, I have a workflow example here for a single study. This is for a modified 1-generation study that was conducted by the NTP as an example. So within this study, you have multiple populations. You've got the parents, you've got offspring, you've got different generations. Within those, you have 48 measurements for all of those populations, or most of those. And then you have different measurements of those endpoints within those studies. Each of those measurements might apply differently to each of our lines of evidence or modalities. So some of those measurements might be predictive of estrogen pathway responses. Some of them may be predictive of thyroid, for example. So we take this data through a number of integration steps for both activity and adversity to help develop conclusions. So one example of this is an endocrine disruption assessment that we did for Oxybenzone. This is a sunscreen active that maybe many of you have heard of. I've just got a screenshot of a poster shown here, but the idea is to show, how much data essentially comes together. So here we had a protocol. We used the EFSA-ECA templates. We looked at activity, primarily using the in vitro, but also some in vivo studies. We looked at adversity. We had a number of in vivo studies. And we also used the epidemiological data. So collectively, we had essentially thousands of endpoints that we were combining in this structure to help think about endocrine disruption potential. Again, was that pathway activated? And thinking about that both in context of hazard and risk. Another example that I thought is helpful to mention in case you're interested is a recent publication looking at ED activity for bisphenol AF. And this was a case study where they were talking about how to apply these systematic review methods. So they followed the same process that I just talked about for Oxybenzone. They used a particular technique for looking at reliability known as SIRAP, which is specific to in vitro and in vivo data. And they looked at, so they had 88 publications. From those publications, they looked at 309 parameters, which were divided into activity and adversity. They combined those or integrated those in 96 lines of evidence. And they developed conclusions for each modality. But also unique to this paper is they really talk about the advantages and disadvantages of using the systematic review approach and using the structured approach for evaluating endocrine disruption potential. My last example here shows a systematic review that we did primarily on the mechanistic data for aspartame, so low no calorie sweetener. So without going into a lot of detail, you might imagine there's a lot of data for aspartame. So what we did is we systematically identified the literature, and then we extracted information by key characteristics. So the idea here is that we already had groups of information. So things like oxidative stress, immunosuppression, immortalization, cell proliferation. So we extracted information into those categories, and then we looked at the reliability and applied a quantitative framework to help us think about the potential for activity along those events. So the right panel of this figure shows how many data sets. So in some cases, we're combining over 100. And then the left side of the panel shows the different types of the potential activity when you combine those. And the different symbols indicate the type of evidence. So in vivo or in vitro, mammalian, non-mammalian. And the idea behind this is to get an understanding of what that data say in relation to activity of events that may be related to cancer. So this may not be the best solution, but this is one solution that we have taken in a number of publications. But really critical then is to think about the linkages of that mechanistic data. So if you do see a molecular marker of oxidative stress, how does that link to what we know about cancer, and how does that link to potential cancer types that we're even seeing in these other evidence types, animals and humans? So you have to take that another step and think about how these mechanistic events might work together. Do they make a biological pathway? And do they link or not link to the types of outcomes that you're seeing in other data types? Okay, so just a couple slides left, but I'm just looking at the time with our 5-minute late start. I'm just going to indicate that, through all of these practices, we really have identified a number of methodological challenges in looking at the data. I was going to highlight some of the challenges in thinking about study validity and the need to look at multiple types of validity in making decisions. I'm going to skip those for today, looking at time. And close with just a few thoughts, is that really the evidence-based techniques have improved the way that we practice risk assessment, but we know we still have a ways to go. We've added structure and transparency, but the field is certainly still adapting. Tools and approaches are still adapting, and we recognize that collaborations are really critical to advancing the field. So it's been really fortunate to be part of the EBTC program that really facilitates those collaborations and helps to advance the use of these techniques in our field. With that, I will say thank you and be happy to answer any questions.
Thank you, Danielle. I have one question if nobody else has a question. So thanks for that overview. There's a lot. I wonder if you could speak to what may also be a challenge in the EB toxicology field, that is a challenge in evidence-based healthcare, and that is the proliferation of systematic review. So now that people have adopted it, there are a number of things that are called systematic reviews that are not, and then there's a lot of duplication of effort. So even within, for toxicology, even within the EPA, as you know, IRIS and TSCA may do the exact same review.
Yeah.
So could you speak to that challenge in evidence-based toxicology?
Sure. And I think largely we are still, as a field, really at the front end of addressing that challenge of what is a systematic review. Are we even, people are adopting it, but maybe they're not fully adopting it or not fully understanding what it is. So I would say as a field, we're really still at the front end. And so, for example, we see a lot of publications that are submitted as systematic reviews, but really wouldn't be considered one. And then in terms of the duplication of efforts, this is also, of course, becoming more prevalent. And it's particularly challenging because we don't have quite the same types of tools to help reduce that. So when we think about Prospero and registration of a protocol, we have a number of protocols registered there, but it's become harder to put systematic reviews of animal evidence on that site. So Circle and some different efforts are trying to help provide platforms to do that. Largely, we're putting our protocols on Zenodo or Open Science, but people don't necessarily know where to find those or look for those yet.
So I have two questions. One perhaps a little less serious than the other. The first is, you didn't give us the most important piece of information, which is how many kilograms of caffeine a day are safe?
400 milligrams.
Okay.
But it's actually protective for males, about 5 cups, 3 to 5 cups a day. So just putting it out there.
Okay, good. The second question, which is a little more serious, is how do you handle, or do you have a framework for handling rare events that may be catastrophic? The 1 in 100,000 aplastic anemia, where you're never going to get enough data to assess the relationship of that event, or it'll be difficult to get that data. The 1 in 100,000 of PML, a brain infection. So do you have methods or techniques or approaches for assessing rare but potentially catastrophic events?
It's a really great question, because in risk assessment, essentially we protect, at that for those rare events and those sensitive populations. So this has been one of those challenges. We don't necessarily need a body of evidence to say that that's what we want to protect on. So you might not even, if there's evidence to suggest a rare event, maybe it's only one study, systematic review might not even be used in risk assessment, because they might just bypass and say that this is all we have and we're going to regulate on this. So the framework is really in the risk assessment framework more than it is the kind of the systematic review. But the other thing that I would say there is that what we're really seeing is a movement towards using a new approach methods, so in vitro methods and predictive methods for potential rare events. And so we're still validating those, but there's certainly interest in using those.
Well, we had one attempt to go in that direction with the EBTC systematic review with rare DILI, drug-induced liver injury. Yeah. So we have a paper on that, and where mechanistic evidence actually had indicated that there might be potential toxicity to the liver, whereas all the in vivo evidence, including smaller clinical trials, did not. So, and we did systematic review on that.
Mm-hmm. Thank you.
We do have a question that came through the chat. How could more people be trained and inspired to conduct systematic reviews? It is very time consuming.
I'm guessing we all have answers to this here.
Get in touch with us.
Yeah, right. Right. So I think I will highlight evidence-based cost collaboration is a great way to get some of that training. Certainly what we found is, digging in is the best way to do it. But the other thing that I would say about it being time consuming is when it's done well and the scoping's done well, we have found it to save time versus take more time than doing something in a non-systematic manner. Let's see, which tools do you use? Okay, well, tools are a great question. There's an SR toolbox. I think it's srtoolbox.org. Katya, you might have it handy since you're typing, but there is no one size fits all. That's my response. We use different tools for essentially the beginning and the end of the systematic review. So for example, we use a lot of Distiller, but there are a lot of screening tools, Swift, but we use a lot of different tools for appraisal and synthesis. And so there's really no one tool that we can say this will this will help you the whole way.
Yeah, so I encourage you to get in touch with us on our website, subscribe to the newsletter. We have a few reviews going, including one related to COVID, European-driven CHOW project, where we're conducting a systematic map of neurological effects of COVID. If people are interested, we're happy to get you involved.
Thanks, Katya. It's nice to see you.
Hi, Danielle. My name is Riaz. Great talk. I have one question that sort of it leaps a little bit off of maybe Karen's question, and you may have touched on it a little bit. I sort of wrote down, there's no standardized approach just yet, but you did mention some frameworks and some guidance on where or how really, how to do a systematic review in toxicology. These guidelines, I assume they also sort of include recommendations on where to search, what types of evidence, should be included. My question is, do you know that these are followed at least, or in some way, or in the field, so maybe there's no standardized approach, but are people generally pretty good about following the guidelines for how to do them, or do they not? Because from my experience looking at systematic reviews and, sort of drugs, at least, they don't really follow the guidelines all that much for some, for harms particularly.
Well, you might not be surprised. I would say it's exactly the same. Of course, I think this will improve with some uptake, but there's also a pretty significant effort on the journal editors' behalf, and actually EBTC is involved in this as well, and a key staff member to help facilitate that. But really, we're seeing a lot of people say they for example, they followed NTP, or maybe they cite Prisma, but the contents really aren't there. And so we still have a ways to go on that. But I'm not sure that there are many clear solutions if we're to look at the EBM world yet.
Could I just add that the National Academies of Science, as Danielle mentioned earlier, and I did, has been asked to review a number of these processes for EPA IRIS, for EPA TSCA. I was also on a committee reviewing a systematic review of toxicology for the procedures or methods for the DoD. So there are different government bodies that are supposed to be implementing systematic review methodologies and are developing handbooks and guidance, and that's part of the question about duplication of effort as well. But there are some efforts to review those and to have some consistency.
I think Karen, that's a really great point, and Bill, that when we look at reviews being done by authoritative bodies, they're much more aligned with these guidelines. Of course, they're actually applying their own guidelines, and I think where we're seeing less compliance is more in publications in the peer review literature.
Yeah.
Thank you.
I think, Taylor, we're at time. Yeah. So thank you very much, Danielle. It was great, and it was great to see you. Nice talk, and nice to see you, Katja. Thanks everyone for joining.
Thanks, everybody.